This is our presentation for Lab 3 Molecules of Life by Emma and Monica. Here's a quick introduction on cells. So cells are the basic unit of life, and the four most abundant elements of life include oxygen, carbon, hydrogen, and nitrogen. And the four macromolecules include carbohydrates, proteins, lipids, and nucleic acids. And they also all contain carbon and are referred to as organic molecules. Some background information on carbohydrates. They are composed of carbon, hydrogen, and oxygen atoms. They are made up of monosaccharides, which consist of simple sugars, and they are also made of polysaccharides, which are complex sugars. They are used as a source of energy, and they also bind using glycosidic bonds. And here's a simple diagram of the chemical makeup of a carbohydrate. Proteins. They are composed of carbon, hydrogen, oxygen, nitrogen, and also small amounts of other elements such as sulfur. Proteins are the building blocks of amino acids. The building blocks of proteins are amino acids, and they consist of an A carbon linked to an amino group and a carboxyl group, which you can see in this diagram listed below of the amino acid structure. And they also bind through peptide bonds, and protein structure consists of four progressive levels, the first one being primary, then secondary, tertiary, and quaternary. Some info on lipids. They are hydrophobic molecules composed mainly of hydrogen and carbon. They are nonpolar and insoluble in water, and they also account for 40% of organic matter in the human body, and they include fats, phospholipids, and steroids. Here are some diagrams of the three major types of lipids. The first one we can see is a phospholipid, which contains phosphate head, glycerol, and two fatty acid chains. Second one is fat, also known as triglycerides, and it consists of a glycerol, three fatty acid chains. And the last one is a steroid, and they are composed of four fused rings of carbon. And here's an example of two steroids, the first one being testosterone, and the second one being vitamin D. For our experiment, we were given four possible foods, which were almonds, olives, fruit juice, and pasta, and we had to determine the identity of four unknown substances based on the nutrient makeup of each, using chemical reagents for testing. So basically, we were given four unknown food subjects, and they were not labeled, and we had to determine which one was which. So we had to determine if unknown substance 1 was almonds or olives, fruit juice, pasta, etc. The first nutrient we tested for was simple carbohydrates, and for a negative control we used distilled water mixed with Benedict solution, and for a positive control we used glucose, also mixed with Benedict solution. And then we got four additional test tubes and filled them with the four unknown food products, added Benedict solution to them as well, and then we placed all six test tubes into boiling water bath using a hot plate inside of a beaker filled with water for about 3 minutes, or until the color changed. So after we observed and recorded the color of each solution, and a positive reaction would be indicated by an orangish reddish color. Here is us adding the test tubes to the boiling water. So first we put in the positive control, then I just put in the negative control, and that is unknown substance 1. And now up to unknown substance 2, unknown substance 3, and finally unknown substance 4. And as you can see, the colors are already beginning to change and the reactions are occurring. So we can see that some have turned orange, some have stayed the same, and some have just, So for our simple carbohydrates test, here are some photos. The before picture we can see is without the Benedict solution, so we can see positive control is clear, negative is also clear, and unknown 1, 2, 3, and 4 are white, with also unknown 3 being yellow. And after we heated them with the Benedict solution added, we can see that the positive control obviously changed to an orange color, negative control obviously stayed the same, and unknown substance 1 and 2 both turned orangish red, which meant that they do contain simple carbohydrates, but unknown substance 3 and 4 remain the same, and they did not change to an orange reddish color, which means they do not contain simple carbohydrates. The second nutrient we tested for were complex carbohydrates, and our negative control consisted of distilled water added with Lugol solution, and our positive control consisted of starch added with Lugol solution as well. So we obtained 4 additional test tubes and added the 4 unknown substances to each, and then we added Lugol solution to the 4 additional test tubes as well, and we observed and recorded the color of each solution. And a positive reaction would result in a color of dark blue or black. Here's us adding Lugol solution to the controls and unknown substances. So here, Monica is adding Lugol solution to the positive control, which we can see instantaneously turned a dark blue. And here's negative control. We're adding Lugols to it, and no color reaction is taking place. And here's unknown substance 1. As we can see, the color is still white or whitish orange, but not the color we're looking for. Unknown substance 2, and here look, immediately we can see dark blue forming, color forming, so this indicates to us that substance 2 indeed contains complex carbohydrates. Now onto unknown substance 3, no color change, not the color change that we want to see, so that means it does not contain carbohydrates, complex, and unknown substance 4, also no color change as well. So here are before and after pictures with the Lugol solution. Before, we can see clear, white, yellow, white, not really that important, but after we add the Lugol solution, we can see positive and negative control. Obviously, positive contains complex carbohydrates, negative does not. But out of the 4 unknown substances, the only one that we see to contain complex carbohydrates is unknown substance 2 because of its color change to a dark blue, blackish color. Now on to proteins. Our negative control consisted of distilled water added with a Bure reagent, and our positive control consisted of egg albumin, which contains protein, and also Bure reagent. So we got 4 additional test tubes and again added the 4 unknown substances to each, and then added 0.5 milliliters of the Bure reagent to each as well. And after 2 minutes, we observed and recorded the color of each solution, and a positive reaction would have resulted in a purple color. Here is a video of us adding the Bure reagent to our controls and unknowns. Here's the positive control, obviously turning purple instantly. And here we go adding Bure reagent to our negative control. And obviously no color change would occur because water does not contain protein. Now we move on to unknown substance 1. And we can see no color change to purple, just green. That's not what we're looking for. Move on to unknown substance 2. And we see a color change, but it is of blue. And now unknown substance 3. No color change that we can observe. And now unknown substance 4. And yes, we do see a color change of purple. Here are before and after photos after we added the Bure reagent. So before we see nothing, and after we can see that the only substance out of our 4 unknown solutions that had a color change was unknown substance 4, which means yes, it does contain proteins. The last nutrient that we tested for was fats, and our negative control consisted of distilled water, along with Sedan reagent and then more distilled water. And our positive control consisted of distilled water and then 3 milliliters of added vegetable oil, because that contains fats, and then Sedan reagent and more distilled water. So we obtained 4 additional test tubes and then added distilled water and then the 4 unknown substances to each. And then we added 0.5 milliliters of Sedan reagent to the 4 test tubes, and then after that we added another 2 milliliters of distilled water to them. And so then we observed and recorded the color change in each solution, and a positive reaction for fats would have resulted in a reddish-orange color. Here are before and after pictures. So before, without adding the Sedan reagent, we can see positive and negative are clear, unknown substance 1, 2, and 4 are all white, and unknown substance 3, it's not, it's yellow and clear. And after we added the Sedan reagent, we can see that out of the 4 unknown substances, the only one that shows an obvious reaction was unknown substance 3, which we can see contains fats. Now onto our results. So after testing for simple sugars, we can see that unknown substance 1 and 2 both contain them, and unknown substance 3 and 4 did not. When we tested for complex sugars, we found that only unknown substance 2 tested positive. For proteins, we can see that unknown substance 4 tested positive while the others did not. And for lipids, we can see that unknown substance 3 tested positive while the others did not. So now with these given results, we have to determine which ones are almonds, pasta, fruit juice, or olives. So here's our conclusion. We concluded that unknown substance 1 was the fruit juice because it contains simple sugars, which also contains glucose. Unknown substance 2 was the pasta, which is also composed of starch and complex carbohydrates. Unknown substance 3 was olives, and which contained lipids, because olives are very fat-filled. And unknown substance 4, finally, was almonds, because almonds are very high in protein, and so was unknown substance 4. And this concludes our presentation on Lab 3, Molecules of Life.